is how we connected, and I can start actually saying that probably I wouldn't have been able to finish my PhD if it wasn't for them and for all the samples that they collected for me. So Sponges, but as I said, first myself. So you can clearly probably hear from my accent, I'm not exactly local. I'm Italian. I come from in the picture is the Strait of Messina. That's where in Italy, basically, I started studying sponges with my master thesis. I'm not a taxonomist, full disclosure, since the beginning. What I've always done during my years studying sponges is basically trying to figure out how they produce some interesting compounds. I will tell you more about later on. But so in Italy in particular, I was farming sponges to obtain biomass for to produce these compounds. And then that's basically that sparkled my love for these animals. So that was the reason for me to come to Ireland, where I did my PhD indeed in, as we were saying, in the University of Galway. But besides all the research activity, I worked between Italy and Ireland on science communication. In fact, I started again in Italy. I worked with WWF and other environmental organizations to promote basically essentially ocean literacy. At the time, nobody called it like that, but that's what it was. And then when I moved to Ireland, besides collaborating with the guys of Search Ireland, I also collaborated with other organizations focused on science communication, like Pint of Science. And I've started my collaboration with the aquarium when with the aquarium as the secretariat of the Irish Ocean Literacy Network, with on a project to promote ocean literacy through music, and eventually I ended up being what I am now, the education and public engagement officer there. So now it's time to talk about sponges. So I start always with these pictures because there are a lot of people thinking that sponges are plants. And I can't blame them because, as you can see, they often look really similar to plants, but they are not. They are animals. They are still debated, even though I report this paper, these diagrams from this paper from 2017 that clearly said that sponges are the so-called sister group compared to all other animals. So it means they are the first animals to appear on the planet. As I still debated, it's a fight between sponges and common jellies, but we don't get into that now because it's not the purpose of this presentation. Just to say that sponges are really, really simple organisms. They basically, their main characteristic is that their body is pierced by a very high number of little pores, which gives actually their scientific names, porifera, that means pore bearers. So all these tiny pores that pierce their body basically draw water inside their body. This water circulates through their canals. The food for the sponges is filtered through in these canals, and eventually, the water gets out through larger openings called oscules. If you have never seen that, hopefully this video will work, and you can see how cool actually it is, the filter feeding system of sponges. This is a barrel sponge that is one of the species. You see, the dye that was spread outside the sponge is filtered very efficiently from the large oscule of the species. But in spite of the fact that these animals are so simple, as I said, they actually have been found to show more than 30 different types of cells. Some of these cells are used to produce the skeleton of the sponges. I'll talk more about that later on. Some other cells line the surface, internal and external surfaces of the sponges, created what we can call, using very simplistic terms, the skin of these animals. Some other cells actually are very similar to muscle cells, and in fact, you don't see them on this picture, but what I'm showing here is that in some lab experiments, they found that sponges very, very, very slowly, but actually can move, can change position. They are not absolutely sessile as we think of them. And then another interesting type of group actually of cell types that the sponges have are called the cells with inclusions. There are many, many different types. What they have in common is that they have some granules, vesicles inside them that contains many different types of compounds. Also some compounds particularly interesting for us that are compounds of pharmacological application that are as I mentioned already. I'll talk more about them later on. And then also many sponges have stem cells and well, have cells that can be considered the equivalent of what we consider stem cells, and that's what give these animals their ability to basically regenerate their body completely, even from very small fragments, which is pretty pretty interesting characteristic of these animals that made them easy to farm, so as we will see later on again. Going back to their filter feeding system, so basically we said how the water goes through their body, but what is that basically filters actively the food for these animals, the food that is organic matter and small microbial organisms mainly. So it's a particular type of cell that is called guanocytes, is the one that you can see there and magnified in this drawing. These are very interesting shaped cells. As you can see, they have a long flagellum, is called, that part in red, that is basically moving, creates the current that attracts the water inside through the pores inside the sponge body and then out again through the osculus. And then this flagellum is surrounded by some extensions of the cells that create like a skirt around the flagellum. And these extensions that are called the microvilli are basically create a sieve that it was actively filters the water. So now I probably get it too much into details, but I would like to show you how actually these cells look like in if you are able to look them with very powerful microscopes, the electron microscopes. So you have in the two larger pictures where you can see specified the flagellum. That's basically our transversal sections of the cells. So it's like if the cells were cut in half vertically. So you can see the flagellum and you can see some bits of the skirt of microvilli around them, whereas the small pictures on the extreme left of the slide shows you a cross section of these cells. So basically, as if these cells were cut horizontally, and you can see the flagellum in the middle and all the microvilli around. You see quite a few of these guanocytes close to each other, because in the majority of the sponges, they are not isolated. They are grouped together in what is called the guanocyte chambers, which makes the filtering activity for these animals more efficient. And this is another type of electron microscope, which shows you the external perspective, let's say, of these cells. So you can see pretty well their microvilli on the top picture, whereas as regards the other picture at the bottom, you can see how they are arranged in these guanocyte chambers I mentioned to you already. Then this is just a fun fact that is to say not all sponges anyway are filter feeders. There are actually carnivore sponges. They have been found in the deep ocean, and you have three beautiful examples on the right hand side, whereas in the left hand side, this is from a paper from one of the first uh, species to be discovered. You see, it looks almost like a carnivorous plant, as it eats the little shrimp as a carnivorous plant would eat a little insect. Now, but yes, sponges can be found in many different types of environments. They can be found in fresh waters, rivers and lakes. So the two pictures, the green and the purple on the left-hand side, those are examples of freshwater sponges. But the vast majority of sponges actually lives in the ocean. And in the ocean, they can be found from the intertidal environment. In fact, you can see there an example of the breadcrumb sponge that can be found often outside the water at low tide. And the opposite, they can be found deep down in the ocean, where they sometimes create massive so-called sponge gardens, like some of them have been found in Irish waters as well. And also they can be found from the Arctic waters, so from very, very, very cold environments through temperate waters up to the tropical reefs, the tropical coral reefs. And on this point, I want to raise another fact of interest, hopefully, is the fact that in a paper in 2013 stated that sponges can actually are good candidates to replace corals because, unfortunately, due to climate change, global warming, corals are badly affected by coral bleaching, and coral reefs are suffering of that because corals essentially die because of this phenomenon. And sponges have been based on studies of the literature, of the sponge literature, and data from prehistorical data. In this paper from 2013, the scientists were basically saying that it's quite likely that sponges are the coral reefs will become sponge reefs in the future because sponges are more resilient, and they actually have even imagined how a sponge reef will look like. But this paper was challenged a couple of years ago by another paper saying that climate change, unfortunately, will cause like dramatic changes in the hydrodynamics of the ocean, which will make potentially impossible for sponges to actually eat because it will affect so badly the circulation of the water, and as a consequence, the circulation of the nutrients, that sponges may not thrive as they thought in this paper. But the same group, or at least the same main scientist of this paper in 2013 found out in 2022 that sponges may not become necessarily dominant if conditions don't allow, but they are definitely prone to recover from much more easily than corals, because they saw during a very hard, the worst marine heat wave up to 2022 in New Zealand, that even though sponges were badly bleached, sorry, and 50% of the population disappeared, they clearly could adapt quite. Sorry for the distraction. I was actually admitting people while I was talking. Sponges can actually seem to adapt to these changes, so they may cope better than other species to climate change. So sponges play a few important ecological roles. So they are first of all a source of food for several different species. We have a list there. They basically transfer nutrients from the through the water column. So they do what is called the benthic pelagic co co coupling. Oh, sorry, by filtering the water. Yeah, they basically transfer nutrients from the water column to the seafloor. And then still because of their filter feeding habits, the fact that they can filter nutrients from the water, they can limit the eutrophication phenomena. So they can limit the explosion of microalgae with all the negative consequences that have can have. They also will talk about this later on. They basically represent a source of an important nutrient in the ocean that is silica, that is one of the main components of their skeleton, and they also offer shelter for types of organisms. In fact, they often live in symbiosis with other animals, like some examples you can see in the pictures there: hermit crabs, brittle stars, worms, and even seaweeds. But sponges, or what is probably more known in terms of the symbiosis that they can establish, are nowadays, in the last 15 or 20 years at this stage, a lot of research has been devoted to understanding the microbial association of sponges, because it has been found out that sponges can host a plethora of very different types of microorganisms, from bacteria to microalgae to even fungi, yeast, for instance, and viruses even. So not all sponges are so rich in microbes, but some of them that in fact are called high microbial abundance sponges can host up to 35% of their biomass, basically can be represented by microsymbionts. So what does that mean? It's these microsymbionts. It's a symbiosis, so it's a mutual. These organisms have get mutual benefits from each other. So they exchange compounds, but it's something. These microbial associations are particular of interest for us as well, because, as I mentioned before, so sponges have been found to be a prolific source of compounds with pharmacological applications, also known as bioactive compounds, that have been seen to have many different types of applications. Indeed, they are like anti-inflammatory, antimicrobial, anti-psoriatic, anti-malaric, and anti-cancer as well. So these are two pictures of sponges that at the time when I was working in the University of Galway, I knew as the only two species that from which compounds, anti-cancer compound, anti-tumor, sorry, compounds that are now on the market were extracted from these two sponges. There might be more now, I'm not too sure, but it's very interesting to know that sponges represent an important source of these compounds, especially if you think in terms of antimicrobial, for the need that we will have in the future for new antimicrobial compounds. And So it's these compounds, it's not always clear if they are produced by the sponge or by the microsymbionts. That's actually what my PhD here in Galway was about, trying to figure out who was the actual producer of the bioactive compounds of some local species. As I say, it's not, I didn't manage to find it out. It's not that easy to know at times, but regardless, it's sponges still these compounds can be extracted from the sponges, and so it's the production of biomass from these animals is of particular interest, and that can be done in different ways. As mentioned already, that's what I was doing in Italy. We can farm sponges in the wild because we can exploit that property mentioned before, that they can regenerate their body essentially. So from fragments of sponge, we can get um, a, complete new sponge. These, in fact, the two pictures on top are different methods that I applied myself during my master's thesis in Genoa. But sponges can be also farmed in tanks in in uh, infrastructures on land, and also cell cultures and cell aggregations can be produced to basically produce new sponge biomass. This production of sponge biomass is fundamental for the science of the sponge bioactive compounds, but actually sponges have been used for a very long time for other reasons, especially the bath sponges. They've been used for, as the name say, they've been used basically to wash ourselves. They're particularly soft and fibrous, so they are well suited for that role. They also been used for painting, for instance. So they've been on demand for a very long time. And in fact, sponges have been collected from the ocean from centuries. The first evidences of that was centuries ago in Greece, and then also in Florida, for instance. That was the collection of sponges was particularly big before this application, to use sponges for as bath sponges, and that was the main reason that pushed towards the farming of sponges, because in certain areas this as often happens, we are probably all well aware of the issue of overfishing that reflected on sponges as well, because some sponge populations were depleted, and so sponge farming became a way to try to solve this issue that we had created. But so the reason why this was just to give a very brief and simple overview of these animals. But the reason why we are here is to understand a bit more about how these animals are identified. As you have figured from the introduction that Rory said, you are not going to leave this Zoom meeting with a clear idea of how to identify all sponges yourselves. It's more to give an idea of the complexity of the topic. As I mentioned, I'm not a taxonomist myself. I just learned the 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 basics of how this, and was enough to realize that is very, very difficult. So why it's so difficult? Because sponges are I wouldn't say, as Rory said, that they all look the same, but it's even the same species or the same individual can look differently at different times. So that's a result of the plasticity of these animals. So their ability or to adapt to the different environments where they live. That again, it's a consequence of their body plan, of their simple structure that make them very, very adaptable. As we mentioned again when we were talking about the future of the sponges with climate change. So some examples. Hope I'm just realizing now that maybe how I arranged this slide may be a little bit confusing. Hopefully, no. So on the left-hand side, you see an example of one of the species that I studied during my PhD that was called Aliclona similans. It's the same species on top and bottom, even though it looks completely different because the two specimens were collected into completely different environment. The one on top was an intertidal area where there was a pretty intense water movement. So the sponges couldn't really develop the clear branches that you can see instead in the other picture from a much more sheltered subtidal area. The other speech, the other pictures refer, the one at the center is the same species, and then you have two additional species on the right-hand side, but these are all cases of sponges where the of the cases in which the same sponge individual was photographed at different times, and it shows a pretty different aspect. And then, oops, sorry, that was fast. So even the same specimen can show different areas of the same different can look differently because of the physiology. Of course, it can be also the case that the animal may be sick, but it can be a reflection of the physiology of the animal in the moment. And then it's also the environment that not just from an hydrodynamic perspective, but for instance, for other reasons like the availability of silica that, as I mentioned briefly before, it's fundamental for the development of the skeleton of these organisms. So this is a case of a sponge that I studied during my master's thesis in Italy. It's called crambe crambe. So what you see in the picture with the black background, the center of the slide, it's the example of the so-called spicules that are components of the skeleton of these animals. We'll get there in a couple of minutes. So if you look at the map there, you see Italy, you see two different locations in Italy. One is in the north of Italy, that's where I did my master's thesis, and the other one instead is where I come from, the Strait of Messina I mentioned before. So Crambe Crambe in the north of Italy, in the location in the north of Italy, as a skeleton made only of these two spicules, whereas in the area where I come from, that's the whole set of spicules for the sponge there, and that's because of the different availability of silica between the two areas. So apparently looks like there is more silica available where in the area in the south of Italy, and that explains this difference in the skeleton of these animals, which clearly shows how these animals can look different from in, in even though it's the same species. So we so I just mentioned that the spicules, and let's talk a little bit more about the skeleton of these organisms, because it's actually the main it's the main element to classify sponges. So the skeleton of sponges is made of fibers, protein fibers, and some small elements that can be considered like the equivalent of our bones, that are the spicules that I just mentioned in the previous slides. So we have the on the left hand side, you can see the cells producing the spicules that are called sclerocytes. And then on the right hand side, instead, you have the spongocytes that are instead the cells producing the spongin, that is these protein fibers that represent the scaffold for the skeleton of these organisms. Not and the spicules are in the majority of cases uh, arranged inside these spongin fibers, but there are exceptions. Like, for instance, the buff sponges I was talking about before, that we use as for cleaning ourselves, they have no spicules. Their skeleton is only made of spongin. That's why we can use them easily as buff sponges, because otherwise we couldn't if they contain spicules, because spicules are made essentially of the famous silica that I mentioned already before a couple of times, or calcium carbonate. And based on the characteristics of these, the skeleton of the sponges are classified. So they are classified in 4 classes. We have the calcarea, which is made of spicules made of calcium carbonate. Then we have the other classes have spicules made of silica instead. But then we have the demosponges, that is the largest class, and it contains also species with no spicules at all, like the buff sponges mentioned already. Then the Hexactinellida are a particularly interesting class. They are found in deep waters. They are sort of aliens and very, very different compared to the demosponge. And finally, the Homoscleromorpha is the last class that was classified in the recent years. It was originally considered to be part of the demosponges, but for some characteristics that are mainly related to their cytology, to their characteristics of their cell, they've been now considered to be a different class compared to the others. And again, some of them are actually skeletonless. They actually have no skeleton at all. So going now to talk a little bit more in details about spicules that are so they are used. The sponges have, if they have spicules, they have different sets of spicules of different. These spicules have very, very different shapes. You see some examples here. So basically, looking at the microscope at these spicules, it's possible to tell species apart. In theory, it's not that simple, because even the same type of spicule that, if you see on the picture on the left hand side, looks very different compared to the others, but it has to be looked carefully, because it can have he may have the same sponge. One sponge can have one type of spicules, And it's necessary to look carefully at them to see their sides, to see if they have particular traits that distinguish them to each other, like for instance, different tips or different shapes, as you can see on the example of these oxias, sorry, on the right hand side, because they are all useful traits to try to distinguish different sponge species. Also, spicules, they can have different dimensions, and they are uh, divided into main groups. There are the macroscleres that are larger in size, and the microscleres that instead are smaller, and they are differently distributed in the skeleton of the sponges. So it's not just the type of spicules to help distinguish different species, but also the way the microscleres and macroscleres are distributed in the sponge skeleton. But when looking at the spicules, at the arrangement of the spicules, is not enough to be able to distinguish different species. There are other ways. One is to look at their DNA. So by extracting particularly genes and comparing them to sequences that are available on databases online, is possible to find out if a particular species is closely related to other and eventually to identify them. Or other ways are basically other ways that can help to identify sponges is to look at their again the cytology we mentioned already, so at their particular cells. So looking at these uh, other characteristics of the sponges is what is called the integrative taxonomy. So it's there are all these other features that can help identify these animals. Some that we have mentioned already are useful in this sense, like for instance, looking at their cells with inclusions that I mentioned before when I was giving an overview of the sponges. I'll give you an example shortly to remind you which ones they were, or also looking at their microbial compositions. Those are all good additional factors that can help distinguish species, because different sponges have different microsymbionts, or also look how the sponges reproduce can help. Because, for instance, sponges can reproduce both asexually and sexually. So it means they can basically generate new sponges by fragmentation, for instance, but they also can reproduce new sponges through sperms and eggs, and so they can produce larvae. So if you look at the diagram on the right-hand side of these pictures, you see that different groups of sponges can produce different types of larvae, or can actually not produce larvae. They can develop directly from the embryo. So all these are additional elements that contribute to the so-called integrative taxonomy, so can help classify, distinguish different sponge species. So I'm going to give the usual case study that I've been giving since my PhD, because as far as I'm aware, this particular group of sponges, the Aliclona, are some of the most representatives of how difficult sponges can be to be identified. So on this slide, you can see some of the species that I was studying during my PhD. And if you look at the sample of speakers there on the right-hand side, the one circled in blue is the type of the main type of species that is found in the skeleton of this Aliclona species. So it's not easy to identify this species based on the speakers because essentially, to quote Rory now, they basically all look almost all the same. So some I show you now a few comparison to show how difficult can, or more or less difficult can be to classify the sponges. So these two species, you saw one of them before already. So the one on top is called Aliclona oculata, and the one at the bottom is Aliclona similans. So if you look at the pictures on the left-hand side, you can see that what you see there are pictures taken at the microscope of the skeleton of the two species. So again, on top you have Aliclona oculata, at the bottom Aliclona similans. So the skeleton looks pretty different. So the speakers are the same, but the arrangement of the speakers I was talking about before is pretty different because the you see in for Aliclona oculata the speakers create this sort of light mesh, whereas in for the skeleton of Aliclona similans there are many more speakers put together to create the mesh. So the fibers containing the speakers contain many more speakers in this case. It's so the skeleton seems to distinguish pretty well these two species. In fact, if you look at the center of the slide there, you see a phylogenetic tree that is basically built using molecular data. It shows the relationships between the different species. So if you focus on the arrows, you see that they bring you to Aliclona oculata and Aliclona simulans that are actually pretty far from each other. So that shows that these two species are quite yeah, are basically not as closely related as others, and the skeleton is a reflection of that. Even though I must say that when we were looking at the skeleton of these two species with Aliclona oculata, it actually depends on where you take the sample to extract the speakers, because the base of the sponge Aliclona oculata that was close to the sediments, it was basically where the sponge was attached to the rocks. It's much more solid than the rest of the sponge, so actually the skeleton of that part of the body was much more similar to the one of Aliclona similans, that is a much tougher sponge compared to it. So it's not even though it's a much these two sponges seem to represent a good example of how the skeleton, the arrangement of the skeleton can help distinguish them. Still, it's not 100% a given. Then another example of two species that we were studying in my lab when I was doing my PhD are the pink one on top is called Aliclona mediterranea, and the purple one at the bottom, Aliclona cinerea. Again, same story as before. You can see how these two species have a pretty similar skeleton. It's again same type of speakers. The arrangement is actually pretty similar. The fibers containing the speakers are pretty thin. These sponges are, in fact, if you look at the tree there, are more closely related compared to the ones seen before. The only reliable way to distinguish them is basically where they are located, because Aliclona mediterranea, as the name said, is a Mediterranean species, whereas Aliclona cinerea can be found here. So here in Ireland, at least, we can have no issues to get confused between the two species, because yeah, Mediterranean is not here. Different is the case in the Mediterranean, because cinerea, as far as I know, can be found there as well. The genus still is the same. Aliclona, 3 different species here now. So the one at the top is called Aliclona viscosa. The one in the middle, Aliclona sarai. Sorry, Aliclona indistinct, and the one at the bottom, Aliclona sarai. So again, if you look at their skeleton, it's very, very similar. And so again, we can apply the rule that the location. So I was doing my PhD in Ireland, and Aliclona sarai is a Mediterranean species. So again, I had no issue in get confused between Indistinta, Viscosa, and Sarai, because Sarai is not here. But between Indistinta and Viscosa, instead, it was more difficult. These 2 species are sister species. They're very closely related. They look very, very similar, as you can see. The only thing that could help, but not really in reality, is that Indistinta is basically only found in intertidal environment. Viscosa is more of a subtidal species, but it's not excluded that it can be found in intertidal environments as well. So these 2 species, more than probably than all the other ones, showed how it's not that straightforward to distinguish different species. So in the work that we were doing in my lab was contributing to try to give elements to classify these species in the genus Aliclona more reliably. So everything we were doing was contributing to what I was mentioning before, the integrative taxonomy of these species. So as I mentioned before, my research has always been on trying to figure out the production of bioactive compounds in sponges, and in Ireland in particular, I was trying to figure out if it's the sponge or the microsymbionts to produce these compounds. So the pictures that you see on the pictures in gray are actually 2 subsets. So the 3 on top represent the cells with inclusions that we have found in the last 3 species that I showed you, like Aliclona indistincta, Viscosa, and Sarai. So you can see these on this slide, these granules that on these 3 pictures were the inclusions of the cells with inclusions that we could find in these 3 species. So that seemed to be a common trait for these species. So again, not something that will help distinguish them in a straightforward way. Whereas the 3 other pictures at the bottom represent the different microbial association of the 3 different species. So it's if you look at what the arrows point, they point at the microbes that we found respectively in Aliclona indistincta, Viscosa, and Sarai. Of course, if you're not familiar with it, if you're looking at it for the first time, you have probably no idea what a bacterium is there, but you can see that the arrows are pointing at things that look differently. So at least what we found is that the microbial associations were different compared to 3 species. So hey, we had finally an element to that clearly show the difference between this species. And also another element that can be considered to contribute to this, again, this so-called integrative taxonomy. So again, trying to find other ways to help distinguish sponge species is to look at the compounds they produce. So for instance, those famous bioactive compounds mentioned before. And again, that seemed to be this is work that our collaborators did, not ourselves, but that also seemed to start showing that chemistry can also be an element to distinguish the species, because the compounds they produce seem to be different com um, compared to each other. So this was an overview of how complicated this can be to distinguish sponges. These are some of the resources that you can find online or you can buy. There's this Systema Porifera is basically the Bible for sponge taxonomists. The World Porifera database is a very important source of information about sponges, and papers, reading papers on particular species is the only way to learn more about them and try to learn how to identify them. But now the last part of the presentation is about the work that we have been done with Research Ireland since 2019 at this stage. So as we said at the beginning, we met because I'm the first one who has experienced the importance of citizen science. Without them, it would have been impossible for me to find some of the species I was studying. But and this initial help for me translated in the development of what we have called the Sponge Project. So the sponges are some of the least recorded groups in Ireland. So the Sponge Project of Research Ireland basically aims at increasing the number, among other things, aims at increasing the number of records for these species. So the Sponge Project is divided in two parts. One is the actual Sponge Project. So basically, if you, when you go diving or when you go walking on the shore at low tide, because as we said, there are also intertidal species, you find a sponge of interest. You are curious, you don't know, you have no idea what that sponge is, and you would like to find out what it is. You could send a sample to us, and we could hopefully help identify the sponges. I say hopefully based on of all the difficulties that I said before, but we have, so the Sponge Project is a collaboration between Research Ireland, myself, who I am now in the aquarium, but still we collaborate with my ex-lab in the University of Galway, in the zoology department of the University of Galway. So if all the procedures to analyze the spicules, the skeleton of the sponges, or if it doesn't work to identify the sponges, molecular techniques can be applied. So we could be able to give you the identification of your sponge of interest. So what do you have to do to collect these samples? So here are the basically the guidelines. So you need to take possibly a clear picture of the sponge in situ, so in the field where you found it. So besides collecting the sample, you ideally should collect as many information as possible about how to describe this sample of this sponge that you want to be identified. So I'll give more information about that in a second. So the samples should be preserved in absolute ethanol, because that's basically what allows, if necessary, to preserve the DNA to carry out the molecular identification. And once you have the your sample, you can send it to Research Island. As regards this absolute ethanol, don't be scared about that. So if you're interested in taking part in the sponge project, we can provide kits with tubes containing it, but we'll get there at the end of the presentation. So going back to examining now each point, when I said, what does it mean, clear picture? Essentially, all pictures like the ones at the bottom can help us identifying the sponge. The one at the top, clearly not, because we can't see the sponge clearly. Then, as I was saying, it's very, very important to collect all the possible descriptive features of the sponge, because the pictures help a lot, but they are not enough, um, because yeah, sorry, I'll get what I want to mean later on. For now, some descriptive elements that you can give is the shape of the sponge. Sponges have very different types of forms. You can see here on the slides, they can be branching, they can have the shape of a fan or a cup, or they can be tubular, they can be encrusting and massive. But encrusting and massive these are very general terms. The sorry, that's an animation that I forgot to remove. That we can go into encrusting and massive. It's very general. If you will give more information about what you mean for encrusting and massive, will be useful. What I mean with that, like for instance, encrusting sponges can be very, very thin, or they can be thick. The encrusting is because they grow like a sheet on rocks, but that the thickness of that can be very, very different for different species. Massive, same. What's the shape of these massive sponges? Like for instance, you can have globular sponges, you can add additional elements, like for instance, if they have a stalk or not, because completely different species, that mean that little information can help to us identifying completely different species. Then still, sorry. Okay, still going back to the encrusting, even just saying thin or thick is not enough. More information would be useful. Like, for instance, the presence of particular character features of the body of the sponges, if they are present. Like in some cases, sponges can have very evident pore sieves. They can have more or less long projections of their body. They can have clear so-called papille, these short projections you see in the picture there. Their surface can be smooth or can be spiky. You can see clearly the canals where the water flows through their body from the surface. That's a very important element to add to your description as well. And you can add whatever you think it's important to describe your sponge. Like for instance, with the last picture there, you can see that they almost look like they have a net inside. That's a type of skeleton that, in my limited experience with sponges, but I worked a lot with these Aliclona species, it reminds me of some of the species I was studying. Then still another important element that you can describe is the color, because as you can see on the slide there, can be diagnostic of related species, like these these two species, the blue and the red, and the yellow as well. They all belong to the same genus, but those are their characteristic colors. And it's same for these species at the bottom. These colors are these species are not related to each other, but their colors are very characteristic for them, so that help them distinguish compared to other species. Then there are other fishes still that you would try to describe using other of your senses as well. Like so, some important features are like, for instance, the production of slime. You can try to see if you can tear a little bit of your sponge if you see some filaments going from one side to the other, as you can see on the picture there, or if you touch the sponge and it feels like it's jelly, so that can be this species produce slime, and that's already an important element that can help identify them. Like, for instance, Rory was saying to you at the beginning that when they were collecting sponges for me, they were often wrong, unfortunately, because it's simply not easy. They were collecting for me a particular species, Aliclona cinerea, that produce these threads of slime, like the one you can see in the picture there, that it's not that easy to see underwater. So they were collecting other species that were very, very, very similar looking to Aliclona cinerea. The only way to be sure that Aliclona cinerea was Aliclona cinerea was to see if there was this slime. So it's actually really important diagnostic feature. Also, the surface of the sponges, as actually mentioned before already, can be very important. So try to touch your sponge to see if it's smooth, because you might not, sometimes it can be clearly visible as it was in the picture before, that is almost, it has a spiky surface, but other times you may need to touch your sponge to be able to describe the surface, the consistency as well. As I gave the example before when I was talking of these two species, Aliclona oculata and similans, one is softer than the other. Similans is pretty hard, so the consistency, it's another important feature that can help in the identification of your sponges. The shape of the oscules, as I've seen before with other species as well, that can be an important element to add to the smell. This is a big one. Is probably not as helpful as other, or definitely not as easy for someone is collecting sponges to describe the smell. But I always like to remember that I was told when I was in Italy that Fatto Russo was a famous chemist who was studying, was actually, or is, I'm sorry, I'm not sure. Is a chemist who was studying the bioactive compounds of these animals, who was able to distinguish different species based on their smell, probably that, yeah, it was smells, maybe given by these bioactive compounds. It sounds like a fairy tale, but in reality, look at that picture there on the slide where it says smell. That's a particular species, Circinea variabilis, that I was studying when I was doing my master's, and the smell was actually different compared to other species. So it is, the smell is actually a serious diagnostic feature as well. And finally, the habitat, as I mentioned before when I was describing the case studies. If a species can be found only in the Mediterranean and not here, clearly that's not, that helps. Or also, as I was telling before about the Aliclona indistincta, that is again the sponge shown there, it can be found, basically, it has been found almost always in intertidal environments, so that says that if a sponge is collected in subtidal environment, most likely it's not indistinct. So the habitat can be important to distinguish species. And so in terms of the sites to collect for your sponge, here on this picture, you have an idea of what would be the ideal size, because it's the first step will be to make spigule slides with spicules, slides with skeleton of the sponges, and it might be necessary to do it more than once. Or if this it's not enough, we may need material for molecular analysis as well. So if it's possible, it's better to collect a good chunk of the sponge to have enough material to do the study. And so another part of the Sea Search Island sponge project, aimed, as we were saying, at increasing the number of records for these animals, is called the Sponge Safari. So this is based on the idea that some species of sponges are very very easy to identify just visually, because there are really no, in Ireland at least, there are no other obvious species that can be confused with them. So the sponge safari is based on 6 species that I'm showing you now. So the first one is the boring sponge, that is called like that because it can basically grow inside rocks. It can erode and rocks and grow inside of them. So in that case, what you see are only these the oscules of the sponges that appear they come out of the rock as this bright yellow papille, but it's often you find the massive sponge that has this very very bright yellow color that is not that common. And again, this distribution, these many papille on the body, uniformly distributed, help identify this sponge that is also massive sponge. And it's this will be the first species. The second one is the black tar sponge, that instead it's an encrustic type of sponge, creates these smooth dark black sheets on the rocks. So it's really also very very clear to distinguish compared to other species. The crater sponge has this lovely light salmon color, and it's called the crater sponge because of these big pore sieves that it does on all along the body. The body is a massive sponge, again, made of these lobes. These pores sometimes can be closed, so that might be difficult to maybe might be not that obvious to identify this sponge in those conditions. But when they are clearly visible, as on these pictures, it's really not that easy to distinguish, to confuse this sponge with something else. Then we have the Purse sponge, that is a type of calcareous sponge, and it's these sponges are actually pretty small. They have they have this very characteristic body shape, that it's like these tiny little sausages, I dare to call them, and on top they have this sort of crown made of spicules. Then the last of the sponges considered for the sponge safari is the chocolate finger sponge, that is called like that because it's a branched sponge that is dark brown in color, and it's not very easy to see this color because talking about the surface of this sponge, it's pretty rough because there are a lot of spicules coming out of the body. So basically, those spicules can trap silt. So often this sponge is covered in silt, so it doesn't look as the name says. But again, also because of this characteristic of having all these spicules on the surface, it's easier to distinguish compared to other species. So these are now the sources to help you getting familiar with the species you can find in Irish waters. And I want now to. I am actually not done yet. I want to show you one thing before, but before we get there, I want to tell you thank you very much for your attention, and saying that if you're interested in taking part in an in-person sponge workshop, you can contact both Research Ireland or myself. You have the email address there. But if you connect to this QR code that you see on the slide there, you can express your interest through a form. We had an in-person workshop in 2019. It goes much more in depth compared to this presentation, of course, because it will be a one-day workshop. And finally, as I mentioned, I want to finish with a video. So to try to promote the knowledge about sponges, the need to increase the records of these animals, and what is the Sponge Project, we have worked with Unbound Media here in Galway to produce a video that I'm hopefully be able to share with you in a second. Bear with me, nearly there.
Maria, I'm not hearing any audio from the video.
Sorry, just a second. I'm not sure why. If you will hear me, I'm not sure why.
No.
Do you guys have the video maybe available, or I may try again. I'm not too sure really what's the issue here.
Groups of animal species in Ireland. Can you hear that, Maria?
No.
Yes.
You look more at home on a tropical reef. Irish waters are home to at least 250.
Yes. So can you guys show the video? Share the video.
Maybe go back that way. But what is a sponge?
Thank you, everybody, for the conversation. Thank you, guys.
It's all right.
Why sponges can be extremely variable in appearance. The majority
There is this. Yeah, right.
Is a share sound button.
Yeah. Take it back to the beginning.
Okay. Take whatever.
Hidden beneath the waves are one of the most colourful and exotic looking groups of animal species in Ireland. while they may look more at home on a tropical reef, Irish waters are home to at least 250 species of sponges, with maybe as many waiting to be discovered. But what is a sponge? Sponges belong to a group of animals called porifera, meaning pore bearer. While sponges can be extremely variable in appearance, the majority of them have the same structure, with the body pierced by multiple small pores from which water is funneled within a series of canals through the sponge's body and out again through one or more larger openings called oscules. This current of water through the sponge body is powered by some special cells lining the inside of the canals, characterized by a whip-like structure called flagellum, which creates the water current. The same cells also have a skirt of cell protrusions around the flagellum, which filter from the water the food particles the sponges feed on. While some sponges are soft to touch as we imagine them, the majority we can see when we dive or snorkel have a skeleton composed of microscopic structures made from calcium carbonate or silica called spicules. Why is recording sponges important? Even though you see them on every dive, sponges are under-recorded compared to other marine species. And marine species tend to be under-recorded compared to land-based species. Sponges can be indicators of marine pollution and the health of aquatic ecosystems, while some sponge species are invasive and can cause damage to man-made structures. Sea Search Ireland is a citizen science project which collects records of marine species in Irish waters and submits them to the National Biodiversity Data Centre, where those records are available to researchers, policymakers, and anyone interested in Irish marine wildlife. Some sponges are extremely distinctive, like the bright yellow boring sponge, but most sponges are hard to tell apart in the water. Luckily, sponges can be distinguished by their spicules, though even then, some species need to be identified using their DNA. This is where the Sea Search Ireland Sponge Project comes in. Divers and snorkelers can take photos and samples of sponges they find at their local sites and submit them to Sea Search Ireland, where the photos and spicules will be examined and the sponge identified for the recorder. These samples will then be stored for future genetic analysis by scientists from the University of Galway. On the shore, you can record sponges through the National Biodiversity Data Centre's Explore Your Shore programme. Divers and snorkelers can take part in our sponge safari by recording 6 common and easily visually identifiable sponges species at their local dive site or signing up to the Sponge Project.